Hi, my name is Shayna, and for my I Wonder research project, I decided to look into the effect of UVA light or ultraviolet A light on plants inflicted with fungal pathogens, or like plants that have fungus basically. And I got to this idea because a few weeks ago, my garden in my backyard actually had a lot of plants with fungus on them, and I decided to buy fungicides to help kill the fungi. But what I noticed was as time was going on, the liveliness of my plants was kind of starting to deteriorate, and my plants were actually looking more dead than they were before I even had fungus on them. And because of this, I wanted to look into different ways that we can treat fungus on plants without having to use harmful chemicals that end up, deteriorating your plant. So before I get into the exact specifics of my experiment, I want to talk about some background information. So right now, a study shows that if 5 of the world's most important plants or like crops, so that would be like rice, wheat, maize, potatoes, and soybean, which are like 5 really popular, I guess, crops that most people include in their diet. And if those are all inflicted with fungal pathogens at the same time, currently 61% of the world wouldn't even have food, which is a really, really great amount. We found that if those 5 plants, and when they're inflicted with fungus, and if those effects of the fungi are mitigated and that the fungi is no longer killing plants, an additional 8.5% of the world would actually be fed. And in a world with 7.8 billion people, that's actually a really great sum of people. And we can even look at history. So like in the Middle Ages, this one fungi called Teleschia really commonly affected wheat fields, and that caused mass starvation really often. So as you can see, these fungal pathogens have a really, really great impact on crop yields and even us as humans. And this kind of led me to think, like, what are some different ways that people have been combating these issues? So obviously, first we have chemical fungicides, and I'm just going to go into the science behind chemical fungicides.
So I looked into the negative effects of fungicides, and so here I have a picture of fungicide, and it's being applied to these plants here. And when we look inside the plant and what exactly is happening when that fungicide is applied. So basically, some of the negative effects of fungicides are that they actually disrupt the role of, or rubisco, which are the proteins, its ability to fix carbon, so carboxylation basically, and over here, so basically, so because that carbon can't be fixed anymore, it can no longer be turned into sugars or like G3P or a part of glucose, and it can't do that, and that part of glucose actually is necessary because plants need glucose and chemical energy to do the work that they need to do. And so because that those fungicides disrupt the ability to do that, the plant ends up actually deteriorating along with the fungi. And another thing that I found was that fungicides, when applied, actually disrupt the structure of the stomata of the leaf. And so basically the stomata of the leaf is basically these little holes in the leaf that allow the exchange of carbon dioxide and oxygen. And because these stomata can't open and close as they're supposed to, there's actually a decrease in the assimilation of carbon dioxide, which is needed to be turned into glucose. And so clearly we see that these issues are very detrimental to the development of these plants. And I was thinking the other day that, is there a way that we can totally avoid the use of chemicals in removing fungi so that these effects, that we no longer see these effects? So after looking around on the internet, I found something called phototherapy. And phototherapy is basically the application of light to an area, an organism that's afflicted with a disease. So in this case, it's fungus. And that application causes the area to heal and the growth of the disease or the fungus to decrease. So here, recently, this method has actually been used in the human medicine industry to combat fungus on human skin. And so as you see in the picture, the person would basically just insert their hand inside this machine that reflects light onto the inflicted area, and that causes the growth of the fungus to decrease and the fungus to die. And so doing that, I wanted to find a way that we can use this method but apply it to plants instead. And of course, the light, how light affects human skin and plant skin is very different. So I had to look into different types of light. And what I found was actually, I found that UVA light actually has um, a good effect on plant skin and also on a negative effect on fungi, which I'm going to get into a little bit later. So basically some background on UVA light or ultraviolet A light. So it's part of the ultraviolet spectrum of light, and it's between its wavelength is 315 to 400. So compared to the other lights in the UVA or the ultraviolet spectrum, it has a relatively longer wavelength, and because it has a longer wavelength, it has lower energy. And so because it has lower energy, it actually inflicts less harm compared to its other ultraviolet counterparts. So, and just one more other thing, that ultraviolet A light makes up 3% of natural sunlight's photons. While researching the effect of UVA light on fungi online, I found an experiment by Maynooth University, and they looked into the effect of UVA light on a strain of fungi called Floridana isolate. And so as I have pictured here, this is just fungi on a strawberry, and I chose strawberry pictures because the way that this experiment was created, it was to simulate the underside of a strawberry leaf, where typically this fungi exists. So basically in the experiment, they plated these Floridana isolates, which are the fungi that they looked at, and they plated two isolates from two different regions, one from Brazil and one from Norway, and they plated them under UVA light in 3 different photo periods. It was after plating it under the light, they counted the conidial spores after 24 hours of incubation, and here's what they found. And so I have a graph pictured here, and so they tested it under 2 different temperatures, and here we have the conidial spores from Brazil and Norway. And so after the photo period, they counted, here's the average number of conidial spores per different group. And so as you see in the dark, there's the most amount of conidial spores, like so an average of 951. And then after 12 hours of dark and 12 hours of light, there's less because we did have that light present. So there's 924. And then in the absolute light, 24 hours of light, we saw only 260.44 conidial spores. So obviously this application of light had a huge effect on the presence of conidial spores. And what this experiment kind of shows us is that the application of light does indeed reduce the germination and the reproduction of these spores, and the application of this light would produce some benefits. And this experiment was significant because it actually took strains from 2 different parts of the world, so like Brazil and Norway, and obviously the conditions of both of those countries are very different from each other, and it kind of shows us that this solution would be able to affect a wide range of populations. In another experiment by the research lab at General Electric, they actually looked at 6 different samples of Aspergillus, which is that fungi that I just talked about at the beginning. And so they plated 6 different samples of Aspergillus, and they put them under different saturations of UVA light from times between 6 hours to 12 hours, and they measured the survival ratio of these spores and the germination time. And so what they actually found was, so here's a graph. So what they found was, no matter the intensity of the light given, there was a downward slope in the survival ratio graphs of each of these different samples. And so it kind of showed us that no matter how much light is being put down, there is an effect on the fungi and their capability to survive. And then in this graph, they graphed the sprouting, so like the germination versus the time, and how long it took to reach that 100% germination, so all of the spores would germinate. And so what they found was, as the light intensity was being increased, the germination time was actually taking longer and longer to reach that 100%. So like the control reached it really fast, and as the intensity was increasing, it was taking longer and longer, and this one actually even just stopped germinating. And so it shows us that UVA does in fact have an effect on the germination of fungi, and it actually can stop the germination of these fungi. So then if we did use this in real life, we would see that there would be a negative effect on the fungi, and that the control of the fungi, we can't establish a control over this. Why exactly did we see these results? So basically the science behind all of this is that when UVA light is put on a plant infected, or it put on fungi, so here's a picture of a conidial fungi, it actually indirectly and directly can damage the DNA and the proteins of these fungi and other biomolecules as well. And so in doing that, the fungi is either immediately killed or the cell cycle is actually halted. So here's like the reproduction cycle on this, and like it typically in one of these steps, it can be halted. And so what this does is that it delays the germination, which is why we saw the other experiments showing a delay in germination, and the population size of the fungi actually decreases. So that's why we saw in the very first experiment, the spores went from like 900 something to 200 something. We've talked about the effect of UVA on fungi. It's time to look at the effect of UVA on plants themselves, because this light would be applied to plants, and we would expect to see some sort of a response in plants as we do see from fungicides. And so this experiment by the Department of Horticultural Sciences and Texas A&M University, they looked at the effect of putting 24 hours of UVA light on 8-day-old broccoli sprouts, which I have pictured right here. And so they measured different compounds within this broccoli sprouts to measure if these plants actually got better or they got worse. And so they looked at glucosinolate, which is right here, and that is basically it's a sulfur compound that appears in really healthy plants. And so I'm going to talk about that substance a little bit later. And they also looked at phenolics, which are benzene compounds in plants, and then they looked at lutein, which is a carotenoid in plants. And after the 24-hour period, they looked at the increase in different compounds in the plant. So they looked at glucosinolate, which I said is that sulfur compound, and this increase is really significant because glucosinolate actually has a antibiotic-like effect in plants. Also, it has that antibiotic effect in humans when we consume. So if we were to have these 8-day-old broccoli sprouts and they had all this glucosinolate, we would actually have it would like affect their antibiotics, or our body's antibiotic capabilities in fighting against foreign substances and viruses. And also glucosinolate is also known to lower chances of cancer because of that antibiotic-like effect. And they also noted an increase in gallic acid and camphorol, and these are phenolics, and so these are like benzene-like compounds that I said before, and they play a role in the development of plants. Like I know that they actually increase pigment biosynthesis and all these other kind of things that are in the plant's development process, and they're very good for the plant. And so we saw this increase for the plant. And then they also looked at lutein, as I said before, which plays a role in plant development. They also noticed a slight increase there. So overall, they found that these really beneficial compounds, like their presence was increasing within these plants, and that helps to prove that UVA light is very beneficial for plants, and applying this as a medication for killing fungi could also show beneficial results in plants. This other experiment actually looked at the growth of lettuce underneath different intensities of UVA. So they looked at lettuce under UVA 10, UVA 20, and UVA 30. And they found some interesting results, which I'm going to go over. So first thing they looked at was dry shoot weight mass increase. So basically dry shoot weight is where they dry and like they put the substance in like an oven to dry it out, so all the water or water is gone, and so that and then they take the mass of that. And so what they found was that under each different light intensity, no matter the intensity, there's actually an increase in weight. So specifically in UVA 20, you saw the most results where you saw around like 29% of an increase, which is really significant because if there's an increase in mass, that indicates growth, so that this plant isn't being negatively affected by this UVA light. They also looked at surface area, and they found that once again UVA 20 exhibited the most results for this. And either way, no matter the intensity, all of them did yield results, but UVA 20 had the most, and it had around 32% of an increase in leaf surface area. And this is really significant because if there's a higher increase in the surface area of the leaf, that means that there's more room for chloroplasts and like more room for photosynthesis to occur in the leaf, in the plant of a leaf. So that means that more chemical energy can be created inside a leaf so that the plant can use to develop and grow. And so, yeah, as you see, no matter the intensity, there was an increase. So that shows that UVA light does have this significant impact on plants and this beneficial impact on plants. And what I don't have this written here, but this experiment actually also found that there was this enhanced secondary metabolite accumulation, and ascorbic acid increased by 47 to 80%, and anthocyanin also increased. And so this was really significant because ascorbic acid actually plays a role in detoxifying the body and fighting bacterial infections when we consume them. So if we were to consume this plant that had this positive effect by UVA, we would be getting these positive effects. And also, I didn't mention this, but ascorbic acid also plays a role in structure of our body, because like it plays a role in formation of different tissues, and hopes to tie in the factor of UVA light killing fungi with the factor that it enhances the development of plants. So basically, UVA light would be applied to plants inflicted with fungi, and then this light would ideally be UVA 20 light, as we saw in the experiment where UVA 20 yielded the best results out of the different lights in the UVA spectrum. And so this light would be applied for around 12 to 24 hours and to a plant with disease. And so this solution is mainly geared towards plants that are inflicted with a conidial fungi that reproduces asexually, as some fungi that reproduce sexually is actually really has not been researched enough, and the mechanisms behind it are still being discovered. So my solution specifically targets fungi, conidial fungi, and some conidial fungi include like powdery mildew, downy mildew, and aspergillus, which are all really, really common fungi that affect crops. So what exactly are the benefits of using my solution, and like how do they apply to the real world? So essentially, the main target of my solution would be the agricultural sectors, specifically in like third world countries, but it can be used anywhere, of course. So because fungal infection can result in a great loss in crop yields, an effective way of combating these losses are very necessary. And so my solution not only avoids the use of chemicals, which can help induce those crop yields, but it also improves the development of those crops, so that people who are consuming these crops are going to be consuming nutritious food. Because typically in developing nations, there's a problem with a lack of nutritious food, and so if my solution can help combat that, that is very beneficial. And it also, because losing crop yields is a lot of money that is being lost, if there's a way to avoid this loss of money, that would also be very helpful. And my solution is also targeted towards everyday gardeners who garden in their own homes and have household plants. So like in 2019 alone, like 33.1 million US households actually garden in their own homes. And of course, fungal pathogens don't really shy away from household plants, and they don't only like attack one sector of human life. So if all these 33.1 million people had access to this solution where they could use, instead of chemicals, they could use phototherapy and a more beneficial treatment, that would be very helpful to them as well.